The first topic is the use of recombinant herpes virus as a vaccine approach for AIDS. And in the second topic, I'm going to talk about viral vector delivery using a viral vector called AAV, or adeno-associated virus, to achieve long-term delivery of the potent broadly neutralizing antibodies that you've been hearing about the last 2 days. And the antibody delivery approach, I'm going to show data on use for both prevention and for treatment. And these two topics are connected by one key element, and that is persistence. Long-term persistence, persistent expression of the proteins that have been inserted that you want to express. You heard from John Mascola yesterday how that there are good reasons to believe that development of an effective vaccine, preventive vaccine against HIV is going to be an extremely difficult task. And I've listed here some of the reasons for believing that. First on the list is that following infection with HIV, virus replication is continuous and unrelenting despite apparently strong humoral and cellular immune responses to the virus. So the natural immune response to HIV, as strong as it is not able to control the infection. Not only is the natural immune response to HIV infection not able to control replication of the infecting strain, that immune response does not routinely protect against superinfection by a different HIV-1 strain. Several investigators have worked on this, but probably the best data is from Julie Overbaugh in Seattle, who has studied a cohort of commercial sex workers in Nairobi, Kenya, and has documented now numerous examples where a person stably long-term infected with HIV-1 has become superinfected with a different HIV-1 strain, and usually that is associated with a jump in viral load upon superinfection. So as strong as the immune response is to HIV, it does not protect against superinfection. So what are we going to put into a vaccine that's going to do better than the natural immune response to HIV? Enormous task. We also have to deal with the issue of enormous sequence variability of HIV. And if there was more time to go into detail, I could show you some figures how drastically HIV overshadows other viruses like influenza for the degree of sequence variability. And finally, and certainly we don't know how to design immunogens that can handle that sequence variability. And finally, we don't know what constitutes a protective immune response, and so we don't even, we're just basically fiddling in the dark. So there are a number of potential advantages for use of herpes viruses or a herpes virus as a vaccine vector. And the first potential advantage that I list here is that the genetic information of herpes viruses is very large and can accommodate large amounts of inserted DNA. And I'll give you more information on that in a few minutes. There are 8 distinct members from which to choose. These 8 distinct herpes viruses have different biological properties, target different cells for replication, target different cells for persistence, and even differ in the catalog of genes that they carry. And then finally, and most important for me, is the issue of persistence. My friend Jay Jung, my former trainee Jay Jung, likes to say, love may be temporary, but herpes viruses are forever. Once infected with the herpes virus, you're infected for life, and the immune responses to that herpes virus persist in an up-on active fashion for the rest of your life. These show the 8 distinct human herpes viruses. These 8 have been can be subclassified into 3 subgroups: alpha herpes viruses, beta herpes viruses, and gamma herpes viruses. I'll point out a couple of things. First thing I'll point out is the varicella zoster virus, the cause of chickenpox in children. There's a live attenuated vaccine for varicella zoster that's in worldwide use. It's given initially as a childhood vaccine, and older individuals, older than me, it's recommended as a vaccine to prevent the reoccurrence of VZV as shingles. To come in with the idea that a live herpes virus is a vaccine, forget it, that's for AIDS, forget it, it's never going to happen. Well, we already have one. Second point that I would point out is that some of the most impressive data in the vaccine arena for AIDS in terms of preclinical monkey studies has been obtained with a simian cytomegalovirus, CMV, by the laboratory of Louis Picker and his colleagues in Oregon. The CMV that they have used is deleted in a couple of key genes, which results in a non-classical immune response to the recombinant SIV antigens encoded by the recombinant CMV. And this non-classical immune response results in 50% of the animals challenged with SIV MAC239, the difficult to neutralize, difficult to protect against SIV MAC239, 50% very stringent control. The virus I'm going to talk about today is a member of the gamma-2 herpes virus group. It's a monkey equivalent of the human Kaposi sarcoma herpes virus, HHV8, which my laboratory discovered a number of years ago. So I've highlighted VZV, vaccine in humans, CMV, impressive results that have received a lot of attention from monkey studies, and the virus I'm going to talk about today, RRV, or rhesus monkey rhabdovirus. And again, I show here, this was initially discovered by my lab, and we've developed a genetic system based on overlapping cosmid clones that forms the basis for constructing recombinant strains expressing SIV antigens. This just shows in red the 5 overlapping cosmid clones that we use for genetic manipulation. So I said how herpes viruses have the potential advantage of being able to accommodate large amounts of inserted DNA. Well, we sort of tried to push that concept to the limit to see if we could insert an entire SIV genome. Okay, and that's the basis for this first part of my talk. We deleted 306 nucleotides that deletes 102 amino acids that constitute the active site of the reverse transcriptase. So this was done, obviously, to obviate any potential for replication competence of the proteins of any virus that might be produced. We also deleted enough sequences from the long terminal repeats at the end so that there would not be one complete sequence of LTR. We inserted this where we usually insert near the left end of the coding sequences of the 130 kb or so of coding sequences of this particular herpes virus. Here I show one of the constructs we made. We put a CMV immediate early promoter enhancer in front of this insert, and we've also used 2 other promoter enhancers that I'll describe here. So this just shows expression of SIV proteins following infection of permissive early passage rhesus monkey fibroblasts in culture. And we have three constructs shown here. One has the CMV immediate early promoter. So we call this RRV, SIV, near full length, NFL. That's not the National Football League. It's a near full length genome insert. We also made a construct with the natural LTR promoter driving expression of the SIV genes, and we used a dual promoter with a mix early and late promoter used by the RRV herpes virus. And you can see these are the proteins for we readily had antibodies that we could detect. You can see the early protein NEF is readily detected in all three constructs, particularly with the CMV promoter driving ready detection of the P24 major gag protein. And all gag proteins are here, and the envelope protein, and slightly higher expression than with the natural LTR and with the dual promoter. So it looks like that this this RRV with the complete genome sequence. Thanks for coming, Steve and Rowena. Welcome. So not only can this herpes virus stably accommodate this 10 kb insert, which is capable of expressing 97% of the SIV proteins naturally, it's making these proteins in the course of infection of permissive cells. Electron microscopy is really impressive, at least for me. If one looks at these infected cells, one can find numerous lentiviral particles, SIV particles, budding from the surface of these infected cells in culture. And this shows the mature particles, SIV particles already budded from the surface of these cells. And some of them have the classical cylindrical or rod-shaped nucleoid, which is a uniform feature of all members of the lentivirus subfamily of retroviruses. These infected cell cultures produce abundant virions by electron microscopy, and levels of P27 core gag antigen with CMV at the highest, LTR in the middle to reflect the protein expression levels, and less with the, this is a control, we don't have the dual promoter here. And you can see over 150 nanograms per ml of the P27, which is what one gets, which is comparable to what one gets in a permissive replication of SIV in culture. So abundant particles and SIV proteins being produced, and we show here that these particles that are produced are not replication competent. The replication here is the positive control. The particles being produced from this recombinant herpes virus are not replication competent as expected because of the deletion of the active site of the reverse transcriptase. So we infected, in initial experiments, we infected 6 monkeys to see what would happen. Is this infectious? Do we get immune responses? The quality of the immune responses. This shows antibody responses to the GP140, that is the external GP120, and the external portion of GP41 following weeks after infection. And you can see very strong responses in 5 of the 6 monkeys. One is weaker, not sure why that is. But the other thing that I'll point out is that not only are the immune responses strong, they persist, okay? And so they don't go up and then fade and go away to memory. This is up on active, lifelong infection with production of good levels of antibodies. Mauricio Martins, in our building, a new junior faculty member, has looked at cellular immune responses and what we've been able to show through this is that there are cellular immune responses to all 9 SIV gene products. So not every animal makes detectable cellular immune responses to each of the 9, but when you look at these 6 animals together, there are cellular immune responses to all 9 SIV gene products. Okay, so we decided then to go forward with a vaccine challenge experiment, and we included 5 recombinant RRV strains in the mix that was used for the vaccine. We used the 3 different recombinant RRV SIV near full length with the 3 different promoters, and we did that because you can do whatever you want in cell culture, but you really don't know how different strains are going to behave in vivo in monkeys, whether some may persist better, whether some may be better at eliciting immune responses because of the nature of the controlled regulated expression. So we included those 3, and we also included 2 constructs making SIV envelope protein alone to try to maximize the antibody responses that we obtained. And we put this into 6 monkeys. Antibody responses, again, persisting and quite good, this time in all 6 monkeys. Oh, excuse me, this shows the anti-RRV response, showing that we started with RRV negative monkeys, and all 6 had a nice, strong, persisting response to the RRV, but they all also had nice, strong, persisting responses to GP140, shown here. This is a tetramer staining in the animals where defined MHC types were used, and again, lots of variability, but nice detection in most cases when there was a match of the MHC type of the cellular response, tetramer responses. So then, so if you know the literature here, particularly from my lab, you will realize when SIV239 challenge is used, you basically have to face the reality of failure after failure. You get your hopes up, doesn't work. You get your hopes up, it doesn't work. You try something new, it doesn't work. So to try to maximize the immune responses here, we included a series of intramuscular DNA electroporations. So we used the same CMV near full length. Someone likes my talk so much they just expressing again all 9 SIV proteins, no RRV, no other sequences, but just the promoter and the 9 gene near full length construct. And you can see here really tremendous boosting effects. And I didn't go out further here, but the persistence of these antibody responses are kind of surprising to me and really quite good. So we've really achieved really high levels of anti-SIV envelope protein responses. And similarly, for tetramer responses, very impressive boosting. So if you look, this is so these are tetramer responses, and following boosting, this animal's over 40% of all CD8 cells are recognizing this one 8-mer peptide. Most of the other animals are lower, but still in a really good range. Actually, in a range similar to what we get with live attenuated NEF-deleted strains of SIV. The ability of these antibodies to neutralize the SIV MAC239 relatively easy to neutralize derivative is shown here. This is prior to the DNA electroporation boosting, And after the electroporation boosting, the neutralizing titers that we're seeing are pretty equivalent to what we see with infection by live attenuated NEF-deleted strains of SIV. The vaccine approach that has far and away protected the best against SIV 239 challenge is live attenuated SIV, and I think second in line is Lewis Picker's recombinant CMV. I've mentioned this a little already, but one of the reasons I've taken this course of research is trying to figure out a way to mimic live attenuated strains in its properties to try to be able to match the protective effects as a vaccine. And here I've shown you so far with this recombinant RRV SIV near full length that the infection is persistent, probably for life. Greater than 95% of the SIV proteome is expressed in this construct. SIV virion particles are formed in abundance. There are immune responses to all 9 SIV gene products, and I didn't show you the data, but the CD8 T cell responses are maintained indefinitely in an effector differentiated state, again, as is seen with live attenuated SIV. So we've completed one challenge, so we're doing repeated marginal dose intravenous SIV 239 challenge, and we did the first challenge 5 weeks ago now, and 3 controls got infected, 3 of the 6, and none of the 6 of the vaccinated animals got infected. The second challenge was done 2 weeks ago, and later this week we'll have the results of the second challenge. I'm excited, thinking that this we may have something here worth pursuing. And for this part of the work, I will thank this first part. I will thank Young Shin, Georg Bischoff, and Mauricio Martins for doing most of the laboratory work, Jeff Lifson for viral loads, and the Wisconsin Primate Center for handling and sampling the monkeys. Okay, and in what time remains, I'm going to talk about the concept of long-term delivery of potent, broadly neutralizing monoclonal antibodies. So antibody-based strategies are made possible by an incredible array of monoclonal antibodies with potent neutralizing activity against a broad range of HIV-1 isolates that have been isolated and characterized over the last few years. And one could think of, as we've heard about repeatedly yesterday, one could think about delivering those antibodies short term by passive administration, and that's okay, but there are some maybe some problems or headaches with doing that. One is the need for repeated injections over prolonged periods. That's going to turn out to be expensive because one has to purify very large amounts of monoclonal antibody for the administration, and one can anticipate compliance issues over those very long periods of time. If one can achieve long-term delivery using a vector such as AAV, there's the possibility of lifelong protection when used as a preventive from a single inexpensive injection. Basically, the goal here is one shot and you're good for life. AAV has a number of distinct advantages. It's ideal in many respects. It has become the vector of choice for treatments of genetic disorders where there is a protein product missing. The only protein expressed from AAV vector comes from the transgene that you put into it. So as long as that transgene product is not viewed as foreign, then one can achieve a very long-term expression. Particularly when intramuscular injection is used. So muscle cells, and I'll show you data that bear on this. So muscle cells basically don't turn over. They live forever. And once the vector DNA gets into those muscle cells, they stay there and can become factories just spitting out the protein week after week, month after month. And it is intramuscular injection that we've used. It's been a proven ability to achieve long-term expression, and I'll show you data from that from our work in monkeys. Outstanding safety record in human gene therapy trials, and as I said, it has become the vector of choice. And there is little or no integration of AAV vector DNA into the host genome sequences, which helps assuage any safety fears. This is basically the vector design. We have usually use the CMV promoter, although we varied that. We've got the heavy chain sequences, F2A self-cleaving peptide preceded by a furin cleavage site that creates the authentic C-terminus of the heavy chain, and then the light chain sequences and the poly-A site. And basically the theme of what I'm going to say here is that when it works, it works. It works with large font, bold type. It works, and it's exciting. And when it doesn't work, which I'll show you plenty of, you want to basically crawl in a hole and change your font to extremely small. So I'm going to first give you the examples of the promise of this approach, results that will illustrate for you why I remain so passionately enthused about this approach. This is one of the first monkeys we did. It received a single administration of AAV1 vector intramuscularly, making the 5L7 rhesus monkey monoclonal antibody, an anti-SIV monoclonal antibody. This monkey has now been stably producing 250 micrograms per ml of this monoclonal antibody for over 3 years, over 3.5 years. And will basically produce this monoclonal antibody for the rest of its life. Approximately 5% of all the circulating IgG in this animal is the monoclonal antibody being produced by its muscle cells from the vector that we inoculated into it. So in terms of trying to envision the promise of this approach, I should also say that this monkey has been protected against 5 successive intravenous challenges with SIV MAC239, including a final challenge with 10 IV infectious doses of SIV MAC239. So this example, I think, illustrates the promise of this approach for prevention purposes. So if we can get delivery like this every time with maybe a couple of antibodies, individuals can receive one inoculation and walk away and be protected for the rest of their life. And these results were described a couple years ago in a PLOS Pathogens publication. Okay, what about therapy? So I'm going to say a few things about our therapy result. So this is one monkey, monkey 2438, that has been dubbed the so-called Miami monkey. We infected it with XIII-AD8 at week 0, and it maintained a viral load set point of approximately 12,000 copies of viral RNA per ml of plasma for 86 weeks, which is a time of recombinant AAV administration. Okay, the AAV1 recombinant that we used, we ended up delivering 2 antibodies successfully, 3BNC117, a CD4 binding site antibody, and 101074, that's shown in green and blue. And you can see levels in the range of 75 to 150 microgram per ml of monoclonal antibody through the 36 weeks here, but we have much later, and these antibody levels have persisted. So this animal now, with those two antibodies delivered, viral loads became suppressed to below detectable levels and have remained below detectable levels for 26 successive measurements over 2 years. One administration, incredible suppression of virus replication. This animal was never on antiviral drugs. We didn't use antiviral drugs at any time. And I'll remind you, 86 weeks of continuous virus replication, lots of diversity, viral load set point around where the median is in untreated HIV-1 infected people, complete suppression. The anti-P27 gag antibodies in this animal have declined dramatically over time since the time of AAV administration. We've done 2 attempts at adoptive transfer where we've taken lymph nodes out of this monkey, made single cell suspensions, transfused it into naive recipient monkeys, and in both cases the recipient monkeys did not become infected. We've made repeated attempts to recover virus from peripheral blood that have been unsuccessful, but but this monkey is not, we now know, is not cured. We have now had 2 successful attempts at recovery of the XIII virus from PBMC using large numbers of CD8 depleted PBMC for virus recovery, needing in the range of 30 million to 50 million CD8 depleted PBMC for virus recovery. So the animal is not cured, but has a very, very impressive suppression of virus. And again, you want to, when it works, it works. It creates this vision of, again, if we can get such delivery every time, delivery of the antibodies, if we can get that result every time, then one administration of the antibody, of the AAV to an infected person, and they can be protected for life, never having to take antiviral drugs again. That's the vision. But as you may have guessed, there are problems, and the single problem is anti-drug antibody responses. That is an antibody response to the antibody we're trying to deliver. I forgot to mention that in all cases we're using rhesusized versions of the human monoclonal antibodies. That is, the constant regions have been converted to authentic rhesus monkey sequences, and only the variable domains come from the neutralizing antibody. So the problem is an antibody response, and this is a slide I used to just show you that. So here we attempted in 4 different monkeys, 1, 2, 3, 4, to deliver the antibody 10E8, rhesusized version of 10E8, which is one of the potent broadly neutralizing antibodies. And in green, you can see the levels of delivered antibody from time 0 when the intramuscular injection occurred. And you can see initially week 1, week 2, nice levels of antibodies starting to come up, but then beginning around week 2, week 3, week 4, an antibody response to the 10E8 antibody, which drives the levels down to baseline undetectable levels. This is the problem that we've been working hard the last 2 years to try to overcome, and it is what is preventing the vision of saving the world from AIDS not becoming a reality. This just shows another example with the authentic 4L6 rhesus monkey antibodies. You can see very nice initial levels delivered for the first 4 weeks or so, but coincident with an anti-antibody response, which I don't show, crashing levels of the antibody in these 6 monkeys. One thing we've showed in that Martinez-Navio paper that I showed the masthead of is that the magnitude of the anti-response correlates directly with the distance from germline. So that is, the further the distance from germline, the greater the magnitude of the anti-anti response. And that's statistically significant both for the heavy chain and the light chain. And that's obviously a problem because all these potent broadly neutralizing antibodies that we'd want to use are highly divergent from germline. They took years to evolve. They've got long CDR3 regions. They're highly mutated from germline. And so this is the basis of the problem. I forgot to say, I'll say it now, that the anti-antibodies are exclusively or almost exclusively directed to the variable domains. They're basically anti-idiotype. They see these highly divergent variable domain antigen combining sequences as foreign, and they make antibodies to it. Is this a problem peculiar to what we're doing, where we're trying to put human monoclonal antibodies into rhesus monkeys, even though we've rhesusized them? And will the ADA problem go away when we use AAV to deliver these anti-HIV monoclonal antibodies to humans? And the answer is no, the problem's not going to go away. So there has been a trial completed by Ivig, International AIDS Vaccine Initiative, performed in England, using AAV1 to deliver the human monoclonal antibody PG9. They got, of the 9 volunteers, they got successful delivery of PG9 in 0 of the 9. And in every case, they had a readily detectable anti-antibody response. So this is not something confined to humans. It's going to be a problem in developing this concept for human use, and we have to figure out a way to overcome it. How am I doing time-wise? I haven't kept track. Oh, I have that much time left? 28 minutes? That's awesome. Oh, 8. Much of the time that we've spent, a good part of the time we've spent over the last year and a half or so, has been doing pilot studies in monkeys to see if we can get some glimmer of hope of a strategy that might help us minimize or overcome this anti-anti response. We varied the promoter, muscle-specific promoter, liver-specific promoter. We've gone large. We've tried using AAV8. So the literature reports that AAV8 expresses well not only in muscle cells but also in liver. Expression in liver is tolerogenic, and quite a number of publications out there have reported that use of AAV8 allows a certain tolerization of the foreign protein that you're wanting to express. We've included microRNA target sequences. These are that we put at the 3' end of the vector. These are microRNA target sequences that shut off expression in dendritic cells and other antigen-presenting cells. The idea being inoculate the AAV, some gets out of the muscle, some gets in dendritic cells, the professional antigen-presenting cells, and that's a major contributor to the anti-anti response. And our colleague at UMass Medical School, Guangping Gao, has done mouse studies to show definitively that's true, that by inclusion of these microRNA target sequences, one can dramatically diminish the immune responses that one gets, at least in this mouse system. The other thing that we tried that I'm going to tell you about is we've tried doing AAV8 as a first inoculation, but then going in with an AAV1, so with the hope being that with the AAV8, even though we may have an anti-anti response in low levels, that tolerization may have occurred, and maybe we can then get good delivery when we go in with AAV1, delivering the same antibodies. I'll show you a few of these. One of the approaches that we're going to continue to use is IVIG. A massive dose of IVIG is used in humans with autoimmune disorders or inflammatory disorders, and for whatever reason it sort of calms or minimizes antibody responses, particularly to self-antigens. And we show here in our 3-monkey pilot that we were able to achieve good delivery of the problematic 4L6 antibody in 2 of the 3 cases, and this was just with a single administration of IVIG at day 1 following the AAV administration. This is the booster that I told you about. So an initial AAV8 inoculation making 4L6 and not too bad levels of expression. And if you look at the if you look at the anti-antibody response, there was clearly an antibody response to the 4L6 that we were making, but that waned over time to low levels, consistent with tolerization. And we then went in at week 50 with a AAV1 making the same antibody, and there was a tremendous boosting. 2 of these animals over 200, over 300 microgram per ml of antibody, and importantly, there was no rebound in anti-antibody responses. So such priming and boosting is a potential way that one can overcome the anti-anti problem and achieve consistent delivery. And this may be particularly useful in a prevention scenario where when our kids get vaccines, it's very often, 2 administrations, 3 administrations, you got to go back and receive booster. So here you go in for the priming with AAV8, you go back 6 months later, 9 months later for the AAV1 boosting, then you can be protected for life. So instead of 1 shot, you're good for life. Okay, 2 shots, you're good for life. So we then started, based on our pilot experiments, we started a new set of experiments. It's very important for me, and it's very important for the field, that we repeat the results with the Miami monkey. The Miami monkey remains 1 monkey, okay? You could argue that, well, the Berlin patient is 1 patient, and look at how the Berlin patient has changed the field and influenced everyone's thinking. But nonetheless, it's important that we be able to get other SHIV-infected monkeys where we successfully deliver these 2 antibodies. So we have 12 monkeys infected with SHIV-88. 6 of those monkeys got the combination of 3BNC117 and 1074 AAV vector, and 6 got an AAV vectors, making 4 antibodies that we've not tried previously: PGT128, N6, PGT145, and 35022. The vector that we used was AAV8. We included microRNA target sequences. We included the LS mutation for half-life, no woodchuck element, and in the back of our heads thinking if we need it, we'll use AAV1 boosting. Here's where we get to the lower font, crawling into a hole. The results that we've got over the last 5 months with this 12-monkey experiment have been very disappointing. This slide shows the one example where we delivered 1 antibody 1074 to 1 monkey, but the other monkeys in this group of 6 had varying levels of delivery, strong anti-anti responses, and by week 8, 10, 12, no delivered antibody. This is the PGT 145 levels, same problem. This time all 6 animals, and early on, nice delivery, 30, 35, 50 microgram per ml of antibody, but then coincident with the appearance of an anti-antibody response, crash, and week 12, week 16, no detectable antibody. And in fact, 35 of the 36 delivery attempts resulted in little or no detectable antibody by week 12. The only one that where we had long-term persistent delivery is this 1 monkey. I haven't given up hope, totally at least. So 2 weeks ago, we did the AAV1 boosting in all 12 monkeys. Each monkey get AAV1, making the same antibodies that they received initially. We won't have, it'll be another 2 weeks before we have initial results on those delivery attempts. And we've also designed new pilot experiments with a few different approaches for minimizing the anti-drug antibody problem. Okay, almost all the work that I described here was done by 2 people in my lab, Jose Martinez Navio and Sebastian Fuchs, who just received his PhD from his work in my lab. Guangping Gao for making and quality controlling all the AAV vectors, Wisconsin Primate Center. All the viral loads were done, Jeff Lifson and his crew at NCI Frederick, and I'll stop there. Thank you very much.
I was just wondering if you have given any thought to, and I don't remember how broad they are, but Julie Overbaugh published a paper on broadly neutralizing antibodies isolated from human infants that I think are much lower divergent from germline in humans.
In infants, because their immune response is not fully developed yet, or infant antibodies, I'm not sure.
They're infant antibodies. So they published a paper a couple years ago where they screened infants that were got mother-to-child transmission for broadly neutralizing antibodies in the infants that were distinct from the mother's antibodies, and they could isolate them and show that they're very low divergent. There wasn't a lot of affinity maturation. I don't really know. I can't remember how broadly neutralizing they really were, but
yeah. I mean, I showed one slide that the as you lower the divergence and get closer to germline, you have less of an anti problem. Guido's Mario Roederer at the VRC has been using anti-SIV antibodies that again are not so divergent, and he sees some of an anti problem, but not as severe as we're seeing.
Gotcha.
But again, I think it's these potent broadly neutralizing antibodies that we want to deliver. Bill Schief is working on ways to minimize the divergence. So, pick here, and eliminate all the changes from germline that you don't need. That's an approach that one needs to go. But at least at this point, I don't see any avoiding the need to deliver these highly divergent antibodies.
So, Ron, why do you think that...
And I would add that this is one of the headaches I've faced the last 2 or 3 years is people saying, oh, this isn't going to happen in humans. It's a problem because it's monkeys. But the IAV results are out now, and it's a problem in humans as well.
So why do you think that priming with one AAV and then boosting with another might get you around the ADA effect? And I have another question after that, if I could.
Yeah, and it has to do with the fact that AAV is 8 is known to express in liver, and for reasons that I don't understand, maybe others do, expression in liver can be tolerogenic, and AAV expression tends to be more tolerizing toward the expressed antigen than other AAVs like AAV1. And I showed you quite clearly in those 3 monkeys that there was tolerization. There was an antibody response initially, it waned, and then we could go back and deliver that antibody in huge amounts, and it was not recognized as foreign.
So maybe the tolerogenic effect of hepatic expression is important. Regarding the difference between muscle and B-cell production of antibody, is there any reason to think that they are the proteins are folded differently or sugared differently or in any other way post-translationally handled differently?
So the antibody levels that we measure in serum are quite accurate because of the reagents that we have, and we've shown repeatedly that the neutralizing activity per microgram of protein is indistinguishable from what we produce in cell culture. So, you know, at least what makes it into circulation is folded properly. We also had a paper.
Hold that. I'm sorry if I can interrupt. I mean, this is really fascinating stuff, but I was wondering with regard to immunogenicity, not so much with regard to activity.
You mean some misfolded?
Some folding that would be recognized differently.
It's possible. But we have a paper in this past year in Journal of Virology where we used RRV to make the same antibodies. And the principal site of persistence, at least in circulation of RRV, is the B cell, just like KSHB. So we thought, okay, we'll use this vector, express it in B cells, and maybe we'll do better. Anti-antis and all that.
I'm curious that you're wanting to use the LS versions of these antibodies. To the extent that you think that this is going to pump out constantly, what makes you want to have the long-acting versions?
Yeah, that's a great question. It's for the last month or so now, I've been arguing with my two people who are working on this, Sebastian and Chema. I'm leaning now toward taking it out. There is a paper that we discovered a month or so ago, publication, and I showed it to John Muscola and to Rick Kaup this weekend, reporting that the LS mutant version of an antibody has markedly decreased antibody-dependent complement-mediated lysis activity, and they're going to go back to see if they've checked that activity with their LS mutants of VRC01. But you're right, do we really need LS? I mean, when we get good delivery, I mean, we're not going to need it. So
it kind of works in with the other part, I guess, of the question. If memory serves, I may be very wrong about this, but I think you generate the LS version by mutating some part of the FC part of the antibody.
correct.
And to the extent that that might make it more divergent from germline, that might, you know, you might solve two problems there.
None of the antibodies that we're seeing are directed to the LS stretch. So we detect no immunogenicity to the LS. Again, it's exclusively or almost exclusively to the variable region.
So, Ron, so in the follow-up experiment where you tried to confirm the Miami monkey, those were SHIV-infected animals.
Correct?
Viremic, right? But
Correct.
did you show what happened to the viral load?
Pardon me?
What happened to the viral load?
Yeah, I didn't show it, limited time, but the animals where we had delivery of 2 or more antibodies at reasonable levels for 6 weeks, 8 weeks, we saw viral loads crash. But then when the anti-antis came up, those antibody levels, the delivered antibody levels went down, then the viral loads went back up. So we don't have any animals of the 12 in this first phase.
Do you think you would...
Where we get 2 or more
Right.
antibodies delivered, and so we don't have any long-term viral load reduction.
Don't you think that the immune system in the context of untreated SIV infection is just a chaotic mess, doing all sorts of stuff and lots of B cell dysregulation and a lot of antibody production, right? We used to diagnose HIV back in the late 80s, early 90s by just measuring their IgG levels because a lot of people didn't want to get tested back then. So my question is, why not put these monkeys on ART, calm things down, build up your response, and then release ART like we do in most of these Curso studies?
You could also argue in the opposite direction. I'm not arguing against what you said. It's perfectly reasonable to be doing in parallel with other things. But one could also argue in the other direction of that, well, if you've got wild-type viral loads and medium-term SHIV infection, there's going to be immune suppression to one extent or another, and maybe being immune suppressed is going to help minimize the anti response that you're going to get. But it's perfectly reasonable to, I think, in the case of HIV-infected humans, it would be very reasonable to do experiments on people who are on antiviral drugs and deliver the antibody with AAV and then try taking them off the antiviral drugs. But there's some hints out there from Michael Farzan and other people that at least one of the antiviral drug mix interferes with the uptake of the AAV. So there's going to need to be a little bit more work to define the conditions for that. But I agree 100% that if you're a firm believer in this approach the way I am and the impact that it can have on the worldwide problem, I would love to see much more of a national effort. I would like to see RFAs, targeted program projects from NIH, to get more people involved in working on this. It's really one problem that has to be solved, the anti-drug problem. And I think when that's solved, it's ready to have a huge impact. And there's only 2 people in the United States that I know of that have grant funding to do AAV delivery of antibodies, and I'm one of them. Okay, thank you very much.
Thank you.
Good.